All right, hi everyone. My name is Brody Fogleman, and today I'll be discussing the paper's scaffolds that mimic antigen-presenting cells enable ex vivo expansion of primary T cells. So I recognize that this is the only paper being presented where a lot of you will have no background. So we'll start with a little bit of background and then go into the results and all that good stuff of the paper. So basically, T cells are just a part of the adaptive immune system and help us fight off invaders. But what exactly is a CAR T cell? So a CAR T cell is basically just a T cell that has been genetically engineered to present an antigen on its surface known as a CAR or chimeric antigen receptor, and it can specifically target cancer cells. So for this therapy, first a patient's blood is collected and filtered to obtain their native T cells, and then genetically engineered to install that CAR on the surface. And then we have to grow or expand the number of these T cells, and then we can reinfuse them back into the donor, and then they'll go in the donor's body and mount an immune response on cancerous cells. So one of the limitations of CAR T cell therapy is the difficulty in expanding CAR T cells into the billions. And this is difficult because the body T cells are activated by other types of cells in the immune system known as antigen-presenting cells. And since a donor that has cancer obviously has an immune system that doesn't recognize the cancer as foreign, we have to expand these CAR T cells outside of the body before we can reinfuse them into the donor in order to be effective. So in the recent years, there have been many innovative techniques used to expand T cells, but many have fallen short because they're so expensive. And as you can see here, in 2019, it was recorded that the approximate cost is $1.5 million per therapy per patient. So beginning in 1977, researchers were beginning to understand how the K562 cell lines could be possibly helpful with leukemia. And then skipping forward to the year 2000, researchers began to use autologous dendritic cells to expand T cells, which are just native antigen-presenting cells. But this proved to be difficult because DC cells are donor-specific and can also result in increased manufacturing hurdles and cost and, of course, time. But in 2014, Butler et al. Introduced the idea that artificial APCs could be created by retroviral infection, and they had limited success with expanding the T cells ex vivo, but this is also really integral to future research. So in 2015, researchers learned that Dynabeads could promote multiple rounds of T cell expansion, and in 2017, the FDA approved CAR T cell therapy to be used for some B cell cancers. And in 2018, which is the year of this publication, it introduces a new scaffold that is better suited to act as an artificial antigen-presenting cell, and it also lowers manufacturing challenges as compared to Dynabeads. So before we dive into the research, it's really important that we understand the three factors that promote T cell proliferation and expansion. And each of these factors that are necessary are shown on the slide, and they work in a specific fashion to promote T cell activation, which can then result in T cell expansion. So the central goals of this paper are to create a scaffold that can better reflect the functions of an APC of the immune system so that T cell expansion ex vivo can be less costly and save time in manufacturing. The second goal is to create a system that can promote both polyclonal and antigen-specific expansion of primary mouse and human T cells, as well as CAR T cells for adoptive cell transfer. And lastly, the authors really want to ensure that the system they create doesn't require purification. So the relationship between genetic engineering in this paper is quite substantial, but not too explicit. So CAR T cells are created by lentiviral gene delivery, and then the cell can produce a specific protein to put on its surface, such as the CAR. So this technology can also be used to design synthetic biology circuits within cells to promote outcomes to confront multiple different diseases, which is what we discussed in class a few weeks ago. And also the authors of this paper really rely on FACS flow cytometry to identify the expression of various proteins and cytokines produced by the cells to understand what is happening at the cellular and molecular level. Without genetic engineering, of course, the creation of these CAR T cells would probably not be possible, and this paper would not be as relevant as it now is today. So the methods that the authors used are very extensive, but I think this figure gives a good overview of the authors' reasoning and thinking about this. So figure A shows the basic steps that were necessary to forming their scaffold that mimic an APC. So basically, the authors created the scheme to make an APCMS, and they cultured T cells on them and looked to see if it would promote T cell expansion. So in the figure on the right, the authors want us to see how their scaffold can promote both polyclonal and antigen-specific T cell expansion. And in the polyclonal animation on the left of figure 1b, you can see that TCR binds to the CD3 antigen on the scaffold, the CD28 binds to the CD28 antigen on the scaffold, and all while the APCMS is releasing interleukin-2, which is essential for T cell expansion. And in the antigen-specific animation on the right of figure 1b, we can see that the TCR binds to the PMHC on the APCMS surface, and the PMHC is just a peptide-loaded major histocompatibility complex, which tells this T cell that there is an invader which is identified by that peptide, and then that the T cell must expand. So the authors formed 6 different APCMS formulations with 4 different parameters. And the next 6 slides will interpret the results from various studies to see which formulation the authors would eventually choose. And I'll pause after we go through the results to see what you think is the best from the conclusions we draw together. And then we'll discuss why the authors chose the formulation they did, and hopefully you'll be able to predict which one they chose correctly and understand why. So there are problems associated with Dynabeads, like we discussed earlier, for the activation, because they do not result in enough expansion and that activation. And this just makes it more time-consuming and expensive. So the Dynabeads are also non-degradable, so they must be separated with a magnet after the T cells are expanded and all that's completed. And this makes the manufacturing process much more complex and rigorous. Okay, so this figure shows the results from a polyclonal expansion study, and figure A shows the images of T cells with the darker spots indicating greater proliferation and expansion rates over time of the cells in different formulations as compared to the Dynabeads. And this can better be interpreted by looking at figure B, I believe, where the results are quantified. So you can see that the fold expansion was greatest in the APCMS formulation A4. And then in figure C, the authors were looking for a stable CD4 to CD8 ratio. So the authors claimed that the APCMS formulation A4 is the closest to this, whereas formulation A1 through A3 showed skewing of that ratio. And then in figure D, the authors looked for 2 markers expressed on the cells that are activated from both the Dynabeads and APCMS formulations. And then from other studies, it's been shown that dual genetic knockout of LAG3 and PD1 enhanced T cell effector function and then delayed tumor growth significantly. So we want to limit the percent of both of these that are expressed. And you can see that formulation A4 of the APCMS did this the best. So now let's look at an antigen-specific expansion of primary T cells. So figure A shows 2 bright field microscopy images, and the authors created the APCMS with either an irrelevant peptide on the left or a known peptide on the right, which was the synfecal, and then cultured CD8 plus T cells with the formulation. And figure B shows the fold expansion of 4 different formulations of the APCMS scaffold, and that formulations A4 and A6 are similar. So in figure C, the authors investigated the expression of interferon gamma for APCMS formulations A3 through A6. And in each case, the formulation was cultured with CD8 plus T cells and pulsed with either no peptide, so negative PEP, or that synfecal peptide, which was the plus PEP, had different effector to target cell ratios. And in this case, the effector cell was the CD8 plus T cell, and the target cells in the culture were the B10F16 cells, which are a type of mouse cancer. So recall that as we discussed previously on slide 12, I think it was, that the major histocompatibility complex must be loaded with that recognizable peptide in order for the T cell to expand via that antigen-specific activation pathway. Also, the negative PEP acted as a successful negative control, just as the authors expected. Okay, so this is the same figure C as was in the previous slide. It's just a little bit reformatted. So it is known that IFN gamma drives activation of natural killer cells and innate lymphatic cells, and it also promotes synthesis of CXCL9 and 10 in the immune cells. So here's a scenario to help get your wheels turning and those light bulbs flickering. I want you to click pause, think about this on your own, try to determine what you would recommend to the manufacturer based on what we've learned so far, and then continue playing the video once you think you have it. So if you recommended to the manufacturer one of the higher E to T ratios, such as 50-1 or 25-1, you would be correct. The higher ratio of effector to target cell indicates that there would be more CD8 plus cells available to attack the target. In this case, that was the cancerous cell. So as indicated in this figure, figure 4E, the higher effector to target cell ratio conditions resulted in higher percent cytotoxicities, which is what we want when fighting cancer. But what about the specific type of formulation that we should use? We'll look at that in the next slide. So you can recall that figure B suggested that formulations A4 or A6 were the best for fold expansion over that 13-day period. Something that you probably didn't catch, though, was the labeling of the y-axis in figure C for the differing APCMS formulations. And this is tricky because it's really essential to what formulation they would ultimately use in the later CAR T cell studies. Since the IFN gamma expression in A4 is higher, as you can see in figures C and D, the authors chose to investigate the CAR T cell expansion studies with this formulation. And figure D is the percent quantification of the CD8 plus T cells that express IFN gamma and IFN alpha cytokines when FACS flow cytometry was used to analyze the expression profiles. And the authors were looking here to see which formulation type had the highest interferon gamma and alpha percentage, which is indicated by the orange portion on that graph, figure 4D. So figure E shows that the higher E to T ratios would be better because they increase cytotoxicity, as we previously discussed. So the next figure, figure 6, shows the in vivo results from using a mouse lymphoma xenograft model. And as can be seen in figure A, the APCMS formulation A4 had a higher fold expansion as compared to CAR T cells that were expanded using the dynabeads. And figure B shows the percent of CAR plus T cells within the model over time and indicates that the dynabeads were more effective in producing a higher proportion of CAR plus T cells than were the APCMS formulations. But the authors said in their paper that this is probably likely due to more efficient expansion of non-CAR plus T cells in the dynabead condition. Figure C shows the ratio of CD4 to CD8 of the APCMS versus dynabeads over time. And the authors also note that the APCMS formulation is better here because they didn't undergo CD4 to CD8 ratio skewing as did the dynabeads. And then figure D shows the outline of this process. So at time point 0, the mice were injected with the luciferized Raju cells in order to make the cancerous model. And then at day 4, the expanded CAR T cells from either the APCMS formulation or the dynabead condition that were, they could be injected and then imaged at day 7. So figure E shows the images of those mice that were included in the study, and the authors report there were no adverse effects observed in any of the mice treated with either the 7-day or 14-day APCMS expanded CAR T cells, and that overall they worked better than the dynabeads. So you can see in figure 6E, one mice still had cancerous B cells detected after 25 days in the 14-day dynabead expansion condition. So figure F shows the quantitative outcomes of this study by comparing the 7-day and 14-day expanded CAR T cells in the dynabeads and APCMS formulation conditions as compared to untreated mock control. And we can see here that over the 35 days that the flux of B cells was measured, the APCMS formulation worked better than the dynabeads in both the 7-day and 14-day expanded CAR T cells in attacking those cancerous cells. So next we'll look at the discussion. So the authors were really confident in their work, and they mainly draw 4 claims from this research. And the first is that the APCMS scaffold shows an advancement that can better promote both polyclonal and the antigen-specific T cell expansion pathways. And it can also show that APCMS greatly increased the expansion of T cells relative to dynabeads without really compromising the functionality of ACT. So thirdly, the authors claim that these results, if they could be translated into common practice, could potentially simplify, standardize, and pose less hurdles for T cell expansion as compared to dynabeads. And then lastly, the authors put a lot of emphasis on CD4 to CD8 ratios throughout the paper, since they believe this to be really important for not only T cell expansion, but also for ensuring that the cells are functional and able to go and attack cancer, which are very necessary for ACT. So this paper has already gotten over 112 citations within just a little over 3 years since publication. And a few of the large impacts associated with the paper include the introduction of that platform technology that could be fine-tuned for a specific microenvironment, and it has changeable degradation kinetics by surface chemistry, which could help us address other disease states as well. So also this paper provides researchers and manufacturers with a much less expensive and time constraining approach to T-cell expansion. And it also increases the amount of time that CAR T cells could stay active in the donor's body and increases the efficacy of ACT. The authors also suggest that this might be a new technology that could be evaluated and used for targeting more complex solid tumors. For example, what if we could, instead of manufacturing these CAR T cells outside of the body, we could inject their scaffold into the body that could continually expand these T cells and target many types of different cancers. So overall, I think this paper has been really revolutionary for the field of CAR T cell therapy, and it continues to be cited in new research today. So I hope you've enjoyed this presentation, and please let me know if you have any questions.